class of drugs that their structures are necessary to memorize for most of the board exams. So here in this video, I like to cover the major classes that includes antibiotics all the way to ophthalmic drugs. First, I like to start with beta-lactam antibiotics. So beta-lactams includes penicillins, cephalosporins, carbapenems, and also monobactams. So all these beta-lactam antibiotics, 4 classes, have something common, that is beta-lactam ring. However, each of them has a different rings are attached to those beta-lactam rings. Like penicillins have a thiazolidine ring, and cephalosporins have dihydrothiazine ring, and then carbapenems have a pyrroline ring, whereas monobactams have just a beta-lactam ring only. So these are the simple direct questions can be possible from those rings that are attached to those or fused with beta-lactams. Here are the most exams they ask you questions like, for example, here is the question about the penicillins. So this penicillin are attached to the isooxazole ring, eventually make this ring and attached groups make the bulky area around this beta-lactam, so that prevents beta-lactamase to bind to them and breaking those beta-lactam bond, which is done by the beta-lactamase enzyme, and that develop the resistance to the beta-lactams. So here there's a question alert about which of the following beta-lactam antibiotic structures and has an isooxazole ring that improves resistance against beta-lactamase. Just guess what could be this penicillin structure. So this belongs to the chlooxacillin. The structure of this drug is chlooxacillin or oxacillins. If you add chlorine, that becomes chlooxacillin. So like chlooxacillins have more improved version of the resistance against beta-lactamase because of these bulky groups attached to those beta-lactam rings. So next, antibiotics are macrolide antibiotics. Macrolides are large molecule cyclic structures, and also their characteristics is their macromolecule, that means large molecule with lactone ring, and then also have a cyclic esters. And this particular structure is about the erythromycin. However, clarithromycin and erythromycin have the same 14-member macrolides ring, whereas the azithromycin is a semisynthetic, which is a 15 members congener with the nitrogen attached on top of it. So therefore, azithromycin is different slightly, or it is a ring number also different, plus also it contains nitrogen atom in the ring, which makes azithromycin also pharmacologically different in terms of their side effect, and also their pharmacokinetics makes a difference. So here is a question alert. What macrolide is methylated erythromycin? Just guess if you attach methyl group onto this carbon, 6 carbon, that gives you clarithromycin. So your answer is clarithromycin. So next, tetracyclines. Tetracyclines have, as the name tells you, have 4 rings, and then also there is a amino group on those 4 positions, which is essential for its antibacterial activities. So tetracyclines include doxycycline, tetracycline, and minocyclines, and most of them are modified here at the ring 6 ring carbon. So here is a question alert about the tetracycline. Tetracyclines undergoes epimerization at 4 amino group. That means the change in this epimer structure happens. That process is known as epimerization. After it becomes epimerization, the drug becomes inactive. So therefore, epimerization can lead to the inactivity of tetracyclines. Next, antibiotics are aminoglycosides. Aminoglycosides are containing glycosides and also polycyclic glycosides, plus also contain aminocyclitol rings. And aminoglycosides, here the question alert, or aminoglycosides are chemically incompatible with if physically mixed with what type of antibiotics. Which antibiotics, if you mix them physically, can lead to the inactive. So answers are beta-lactam. Beta-lactams and aminoglycosides should not be mixed into one vial or one injection. Next, fluoroquinolone antibiotics. This is the quinolone core structures, which consist of the ketones and also acid groups, which is site where it binds to the DNA gyrase, or also known as topoisomerase 4 site of the binding. So these are the common core structure of quinolones and also contain fluorine on top of it. However, fluoroquinolones are primarily inhibit bacterial DNA synthesis, which target the enzymes such as topoisomerase 2 and also DNA gyrase. However, because of the structural chemical structures, fluoroquinolone known to have a class side effects. So what are those examples of fluoroquinolone class side effects that can be explained from these chemical structures? That is, mainly it causes the QT prolongation and also known to have phototoxicities, and can also have a CNS side effects because some of the structurals have a similarity to GABA, and therefore it can result into peripheral neuropathy, and also it changes the blood glucose level. Sometimes it can result into hypo or hyperglycemia. Next, we have the antifungal drugs, and the antifungal drugs are several classes. However, I picked up the two major classes here. One is imidazole ring, which is a ring with the 2 nitrogen, that is the imidazole, whereas here triazole, which contain 3 nitrogens. So these two major types of azole antifungals, those includes the imidazoles are clotrimazole and miconazole, whereas triazoles are fluconazole, itraconazole, and voriconazoles. Here we have antiviral medicinal chemistry. Among them, herpes antivirals, well-known antivirals, especially valacyclovir. Valacyclovir is a prodrug because it attached to the valine group, and this valine group further, after absorption, it will be detached from this and then makes the active drug that is known as acyclovir. So acyclovir is, here is a question, valacyclovir is a prodrug of acyclovir. Next, you have integrase inhibitors, which are used for HIV infections. So this is the example of dolutegravir. The suffix, the name of these integrase inhibitors are end with the tegravirs. And this site where it binds to the integrase enzymes. And this dolutegravir, also this class of drugs, also known as integrase strand transfer inhibitors, also known as INSTI. And then the purpose of this integrase enzyme is it binds with the viral DNA and integrates into the human DNA. So therefore, that is the integrase enzyme is essential. So the integrase question alert here is integrase enzyme is essential for what? For retroviral DNA integration into host DNA. That is the purpose of integrase enzymes. So let's look at another important class of antiviral drug, which is known as NTRTI, and which is best example is tenofovir alafenamide. The active drug of this is tenofovir. There are two salts of tenofovir in the market. One is the tenofovir alafenamide and also tenofovir disoproxyl fumarate. So here I'm presenting tenofovir alafenamide, which is the most popular one, which is a prodrug, and it will become active drug, that is a tenofovir. And thus, this tenofovir alafenamide is a prodrug of tenofovir. And the more important thing is TAF, or tenofovir alafenamide, is more effective than the other salt, which is a TDF, tenofovir disoproxyl fumarate. So TF induces higher level of tenofovir diphosphate and requires in decreased dose compared to TDF for this class of medications. Next, we talk about the important class of drugs, or oncology medicinal chemistry. So among the oncology medicinal chemistry, there are several classes of drugs. The first, I like to start with the alkylating agent. They're also known as DNA cross-linking agents. They alkylate mainly to the DNA of the cells, so that way they prevent DNA replications. So there are alkylating agents are mainly based on 3 major nucleic acid bases, which is adenine, guanine, and cytosine. Adenine structure is here, and there is a guanine, and then cytosine structures. So primarily these drugs, these adenine guanines drugs-based alkylating agents, or primarily they do contain strong nucleophilic groups. So therefore, these nucleophilic groups tend to make a complex, which is a covalent bond with the DNAs, cell DNAs. So, for example, the alkylating agent cross-linked to the DNA nucleophilic sites by forming strong covalent bonds. So, one of the best example of the alkylating agent is cyclophosphamide. This cyclophosphamide structure is here, and which is a alkylating agent which cross-linked to the cell DNA. However, one more important significant point of cyclophosphamide is known to produce hemorrhagic cystitis, which means bleeding from the urine and the bladder irritation. And that bladder irritation caused by a drug metabolite of cyclophosphamide that is known as acrolein. This acrolein produced those bladder irritation. So therefore, there's another substance called mesna, which is a simple sulfur-containing product, which is known to bind to this acrolein, and that makes the complex, and this makes the water-soluble complex, and that gets out of the bladder and remove this acrolein from the bladder. Because acrolein is a bladder irritant that is known to cause those bladder irritation or hemorrhagic cystitis side effect or bleeding from the bladder. So here is the question alert. So acrolein is a genotoxic and causes hemorrhagic cystitis, which can be treated by what substance? Guess what is the answer here? The mesna, which is remove those acrolein by making water-soluble complex. All right, so other important class of anti-tumor antibiotics or anthracyclines. Anthracyclines are known to have a chemical structure which is similar to tetracycline. However, the difference in the structure will be different here, whereas tetracycline has amines. Here you don't have amines, and therefore, but there are structural similarity. However, anthracycline anti-tumor antibiotics are known to form oxidized product. So basically, first undergoes NADP reductase and then produces this oxidized product. And this oxidized product form those superoxide. And this superoxide is known to produce toxicities. So therefore, superoxide and this mutase enzyme basically that causes the breakdown of superoxide to hydrogen peroxide, and then further catalase enzyme breakdowns to water and oxygen. So that way, these superoxide desmutase reduce those toxicities because anthracyclines are known to produce cardiotoxicity. That's a big point for the exam. So keep in mind every point I'm discussing here that has serious, important significance in the exam's questions. Questions could be as expected here, presented in the question alert, or you may expect question indirectly or directly from these key points. Next, we talk about purine antagonists, antimetabolites. Mainly, there are thioguanines, mercaptopurines. They have something common. They do have a structural similarity. Only difference here is thioguanine, which is a DNA base, which has a NH2, whereas mercaptopurine have no NH2. So these drugs are known to, the key point here is thioprine and 6-mercaptopurines are prodrugs. That has to be become monophosphate by cellular enzyme. So what enzyme is that? That's the key point that is expected in the exam. So the question here is what enzyme that plays a crucial role in metabolism of mercaptopurine, and which is an anti-cancer drug. So the enzyme is known as thioprine methyltransferase. That is a very important enzyme necessary for its metabolism. The other important antimetabolites are methotrexate. Methotrexate is very unique structure, which is very similar, structurally similar to folic acid. Mainly difference here is NH2, which is not there in folic acid. And there are two major parts of this. One is the pterine portion, which is primarily bind to the dihydrofolate reductase enzyme, which is what methotrexate inhibits. And then also there is a glutamic residue, and also there is a third piece here is called PABA, para-aminobenzoic acid. So these three units combined is a methotrexate structure, which is very similar to folic acid. So here, a mechanism of methotrexate is dihydrofolate reductase inhibitors. Basically, folic acid turns to dihydrofolate, by the enzyme dihydrofolate reductase. There are twice this dihydrofolate reductase inhibit formation of it's necessary to form tetrahydrofolate and then also complex of methylene tetrahydrofolate. Whereas methotrexate inhibit this DHFR, which is a dihydrofolate reductase enzymes. So let's look at this question here. Terine portion is active site, which binds to the dihydrofolate reductase. Next important class of drugs or cardiology class of medicinal chemistry. Among them, first let's start with the anticoagulants. Heparin is very unique structure, which is mucopolysaccharides, which contain sulfate. This heparin sulfate make them more acidic. So these sulfates are unique for heparins. So here is the question for this one. Heparins are glycosamine glycans, sulfated polysaccharides. So if you have to say in one word, which is the glycosamine glycan sulfated polysaccharides. Next, you have a factor 10A inhibitors, which is also known as DOACs, direct acting oral anticoagulants. So among them, the important class is factor 10A inhibitors, which is known for rivaroxaban, apixaban, etc. So here, a structure of rivaroxaban consists of three major units. And then structurally, I'm not hoping big question on them, but just to know that rivaroxaban or factor 10A inhibitors, they are selective factor 10A inhibitors. So next we talk about the thrombin inhibitors, which is another DOAC, which is a direct acting oral anticoagulant. The best example is dabigatran, and this dabigatran structure, which has the similarities to the apixaban. However, structurally, the most important key point is this is a different than apixaban mechanism. This directly bind to the factor 2A, which is also known as thrombin. The another name for factor 2A is thrombin. Next, we have the old anticoagulant, that is vitamin K factor inhibitors, which is the clotting factors. Now, warfarin. Warfarin is one of the oldest oral anticoagulants. Warfarin exists as a racemic mixture into R and S forms, this site, and the ring name of this warfarin is called coumarin ring or coumarin. This ring is known for coumarin ring. So here is warfarin is R and S. However, S-warfarin has a more extensive metabolism, which is in the liver, which is a substrate of 2C9, 2C19, and 3A4. Because of that, warfarin is known to have a lots of drug interaction. This is one of the highest drug interaction drug. So therefore, that is highly potential expected question on there. Next, other cardiovascular drugs are angiotensin converting enzyme inhibitors, which is our antihypertensive, which is in short also known as ACE inhibitors. ACE inhibitors have a typical peptide structures. However, it is important for angiotensin converting enzyme. The angiotensin converting enzyme consists of a zinc metal ion. So these free acid groups are essential in order to complex with zinc. So therefore, ACE inhibitors, they bind to the zinc. Like some ACE inhibitors have a carboxylic acids like silosapril, lisinoprils, whereas captopril has a sulfhydryl group in this place, whereas fosinopril has phosphonic acid. So these are the unique structural activity for angiotensin converting enzyme inhibitors. Next, another antihypertensive, which is angiotensin receptor blockers, also known as ARBs, which has a unique structure. For example, valsartan. Sartans have the three major active sites. One is the tetrazole ring, and then you have imidazole ring, and then you have a CH2 ethanol group attached on top of the imidazole ring. So ARBs are analogs of imidazole ring attached to this group that is essential for its activity. Next, beta blockers, which is one of the older drugs. Beta blockers have a typical structures, which is consist of aryl propanolamine with aromatic rings or aliphatic rings attached to them, make them different types of beta blockers. So aryl propanolamine is essential group for beta adrenergic activity. Next, we talk about the important class of antihypertensive, that is dihydropyridine calcium channel blockers, which is also known as DHP calcium channel blockers. So which contains a very unique ring for 1,4-dihydropyridine ring. This ring is essential for its activity. So this is a common structure of all dihydropyridine ring, whereas there are substituents that are attached to them or various different groups make them different types of dihydropyridine calcium channel blockers, which is like amlodipine, nifedipine, felodipines, etc. So here, dihydropyridine calcium channel blockers contain 1,4-dihydropyridine ring. And also they undergoes hepatic metabolism, which is therefore they do have a serious interaction with CYP3A4 inhibitors. Next, the older class of antihypertensive thiazide diuretics, which is well example, is hydrochlorothiazide, which consists of sulfonamide. As a result of that, that causes sulfa allergy. And then also there are small changes in those rings here. However, in this one, there are examples of thiazide-like diuretics. Chlorothalidone, metolazone, and indapamide is known for thiazide-like diuretics. Very important class of drug in cardiovascular drugs are HMG-CoA reductase inhibitors. In short, it is also known as statins. For example, lovastatin structure is here. So all the statins have this HMG-CoA reductase intermediate structure. So this is essential for most statin structures. So that is also known as 3,5-dihydroxycarboxylate, which is or essential for statin structures. Next, other antihypertensives, other drugs of the cardiovascular include antiplatelet agents. Among them, the promising agents are P2Y12 inhibitors, also known as ADP inhibitors. The best example here is a clopidogrel. So the clopidogrel is contain thienopyridine ring system, which is a unique system. And then this clopidogrel also has serious drug interaction because it is prodrug. It is metabolized by various enzymes, and that one of them is CYP2C19. As a result of that, these CYP2C19 inhibitors, the major examples are proton pump inhibitors. They're known to cause those drug interactions. So next, we have analgesic and anti-inflammatory drug classes that include NSAIDs, which is a major class of drug. Among them, the most popular ones are brufen. Brufen is a racemic mixture, and which is, however, S-brufen and R-brufen, whereas naproxen, which is a long half-life, which is a pure S-naproxen. So in case of brufen, you have to know one thing. Brufen is a racemic mixture. However, activation of carboxylic acid here is stereoselective reaction. What that means is S-brufen is much more active than R-brufen, whereas naproxen is a S-selective enantiomer. Next, we have opiates. Opiates chemistry has been studied a lot, and therefore there's always potential questions. Among the opiate structures, there is something 4-phenylpiperidine ring, which is essential for its activities. So all opiates contain that rings. So natural opiate, the best example is morphine, and most of these natural opiates have been semi-synthesized. Like for example, opiate morphine modified here, alcohol to ketone. Those small modification here to this, and that makes semi-synthetic opiate, which is a hydromorphone. Hydromorphone is way higher potent than the natural morphine. So here is a question alert. The reactive metabolite of morphine is morphine 6-glucuronide. This 6-glucuronide is necessary. It has more activity than original morphine. Next, antihistamines are known for very popular chemical structures, and among them antihistamines, ethanolamine antihistamines, which is known for first generation. First generation antihistamines known to cause more sedation side effect or drowsy side effect, just because that consists of tertiary amine ring. This tertiary amine group makes them more penetration into the brain and cross the blood-brain barrier. Plus, also structurally, they have more lipid-soluble structure, so that enables more drug passing into the brain. As a result of that, results into more sedative, drowsy side effect of first-generation antihistamines. Next, we look at the endocrine medicinal chemistry. Among them, antidiabetic drugs, especially the metformin. Metformin has a unique structure, which is known as biguanide class of structure, which is primarily this structure here undergoes a process known as tautomerism. Tautomerism means you could see there is a shift in the hydrogen ion from one nitrogen to other nitrogen. That process is known as tautomerism, which is called a reversible migration of hydrogen atom between the two nitrogen is known as tautomer process. Next, the new class of drugs are SGLT2 inhibitors, such as gliflozins. So SGLT2 inhibitors are very unique chemical structures because they do have a C-glycoside link attached to those benzene ring. So here is the example, empagliflozin structure. So SGLT2 inhibitors have a C-glycosylation, which is essential for SGLT2 activities. Next, another class of drugs are DPP4 inhibitors, which is end with the suffix called gliptins. Here the structure of sitagliptin, which contain quinazolinone ring or cyanopyrrolidine-based structures. Next, other hormonal drugs are adrenal corticosteroids, which is two types of them. One is aldosterones, hydrocortisone. Aldosterones also known as mineral corticosteroids, whereas hydrocortisones are known for glucocorticoids. So the mineral corticoids, aldosterones, have a very typical structure which contain aldehyde group on this carbon, which results into formation of acetal group. Aldehyde group equilibrium and form this a four-member ring or five-member ring between here, and that is known as hemiacetal. So whereas the glucocorticoids, obviously they have methyl group here, and for both of them, the 11th position hydroxyl groups are essential, or ketone groups are essential for their activities. So next here, the question alert is the mineral corticoid aldosterone has aldehyde equilibrate as a hemiacetal. Next, gonadal hormones, the best example are estrogens. Estrogen contain phenolic ring, and then also the synthetic estrogen, which is also known as ethinyl estradiol, would contain ethinyl group. And this ethinyl group makes this synthetic ethinyl estradiol is a more orally stable drug. So that means oral preparation. That means oral contraceptives. Most of them are made with ethinyl estradiol synthetic hormones, estrogen hormone, which is because you attach this ethinyl group. Whereas natural estrogen does not contain, just has simple hydroxyl group here. So here is the question alert. So what is that happens because of the phenolic ring? Phenolic ring in the estrogen causes carcinogenic side effect or cancer, which is one of the breast cancer side effect, which is a complete contraindication of estrogen that is coming because of this phenolic ring. Next, other important class of drugs are neurology drugs that include serotonin, dopamine, norepinephrine. These are the three major neurotransmitters. Serotonin is indole ring, contain indole ring, whereas dopamine and norepinephrine, or both of them, are catecholamine type of neurotransmitters. So these are two are called catechols, which is basically makes because of two hydroxyl group. Whereas this ring here is the indole ring that is present in serotonin. Next, here we talk about the anti-seizure medications. Anti-seizure medication, there are lots of them. However, carbamazepine, made of dibenzodiazepine. Most important point about carbamazepine is its drug interaction, because carbamazepine is a potent inducer of what cytochrome enzyme. It is a potent inducer of CYP3A4 and also other cytochromes, plus also phase 1, phase 2 enzyme system in the liver. Important class of anti-seizure drug is valproic acid. Valproic acid undergoes several metabolic reactions. One metabolic reaction of the metabolic reaction of valproic acid is oxidation. It makes two hydroxyl groups here, and one 4-hydroxy, other one is 5-hydroxy. So question alert is, what is this 2-hydroxyl groups or types of isomer that is known as structural isomers? However, valproic acid also has a very famous glucuronidation phase 2 metabolic reaction, and that glucuronidation also results in a serious drug interaction with lamotrigine. That's also important point. All right, so next we have another anti-seizure drugs, which is barbiturates and phenytoin. They do have some structural similarities. For example, here the structure of phenytoin and phenobarbital, they do have something similar. So therefore, they do have something common. That is, phenobarbital and phenytoin are structurally similar, and they do have a CYP3A4 enzyme induction. They are very famous enzyme inducers similar to carbamazepine. Next, benzodiazepines are very well-known drugs. Benzodiazepines have a common structure that include the ring B. The common ring B is present that is known as diazepine ring. The next, similar structures. However, they do have this alcohol, and this is not directly attached to the benzene ring. However, this group is essential for its activity. So here, that is known as N-tertiary butyl group, makes it more selective to beta-2 agonists, whereas long chains here are present for long-acting muscarinic and long-acting So this is a mistakenly written here. This is not a SAMA LAMA. This is a short-acting SABA, and also this is a LABA, which is a short-acting beta agonist, and then long-acting beta agonist, which is beta 2 agonist, not the LAMA. Next is about that anticholinergic drugs. So all anticholinergic drugs, they do contain a quasi-ring, that is a combination of two rings called pyrrolidine and piperidine ring. And there are SAMA, short-acting muscarinic antagonists, ipratropium, long-acting muscarinic LAMA, that is known as tiotropium or glycopyrrolate. There are more drugs belongs to this class. Next, we have a gastrointestinal drugs class, especially one thing I recommend about omeprazole, which is a racemic mixture, whereas S-omeprazole is a pure S isomer. So therefore, that's the structural same, but they are optical isomers, whereas S pure isomer is S-omeprazole. And then other one is misoprostol. Misoprostol is known as eicosanoid, which belongs to the prostaglandin E1 analog, which consists of ketone and hydroxyl group, and there's another hydroxyl group is present. So misoprostol is what? Here is a question. Misoprostol is eicosanoid. Chemically is known as the structure misoprostol, but they are not exactly same. There you could see there are two hydroxyl group instead of ketone on the 9 and 11 position, whereas previously it was ketone here, but now here this is hydroxyl group. These are known as PGF2 alpha analogs, right? PGF2 alpha analogs. These are prostaglandin